Center for the Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on bacteria in industrial processes. In our last lesson, we studied the use of bacteria in the production of certain food and beverage items. Products like injera, cheese, yogurt, te'ella, edge, and beer are made with bacteria through fermentation. We learned that fermentation is the process by which bacteria and yeasts break down carbohydrates to form alcohol and carbon dioxide. Today, we will continue to look at the ways bacteria are used in industrial production. We will discuss how bacteria are involved in manufacturing antibiotics. Let us get started with the lesson. Antibiotics are chemical substances that can inhibit the growth of, and even destroy, harmful microorganisms. They are made from other microorganisms and are produced on an industrial scale using the fermentation process. The first antibiotics came from fungi. Today, more are being made with genetically modified bacteria. Students, have you ever used antibiotics? For our first activity, please take a few minutes to write down your experience of using antibiotics in a sentence or two. Describe your illness and what the antibiotics did to treat your illness.
Students, let's get ready. Begin. Time's up! Let's get back to our lesson.
Welcome back, students. I hope you were able to complete your sentences. Antibiotics are used in many forms and to treat many different infections. Each application of antibiotics requires a different method of manufacturing. For bacterial infections on the surface of the skin, such as an eye or ear infection, the antibiotic may be applied as an ointment or a cream. If the infection is internal, the antibiotic can be swallowed or injected into the body. Antibiotics differ chemically, so it is understandable that they also differ in the types of infections they cure and the ways in which they cure them. The development of modern antibiotics was based on the work of a few individuals. These scientists demonstrated that materials derived from microorganisms could be used to cure infectious diseases. In 1928, Alexander Fleming made one of the most important contributions to the field of antibiotics. In an experiment, he found that a strain of Penicillin mold inhibited the growth of bacteria. This led to the development of the first modern antibiotic, penicillin. Penicillin is one of the most important discoveries in medical science. It is a commonly used drug, and it led to the development of many other antibiotics. Although most antibiotics occur in nature, they are not normally available in large quantities. The fermentation process involves isolating the desired microorganism. Next, the microorganism is cultured and put into a fermentation tank to yield antibiotics. The antibiotic product is purified and isolated from the mixture. Lastly, the antibiotics are refined and formed into the various product types. It is important that sterile conditions be maintained throughout the manufacturing process, because contamination by foreign microorganisms will ruin the fermentation. Let us discuss the steps in the fermentation of antibiotics in more detail to better understand the process. The first step is starting the culture. Before fermentation can begin, the desired antibiotic-producing organism must be isolated and allowed to grow. A starter culture in an agar-containing plate is used to begin this step. When the culture is ready, it can be transferred to a seed tank designed to provide an ideal environment for further growth. The culture is supplied with all the things the specific microorganism needs to survive and thrive. This includes warm water, carbohydrate foods like lactose or glucose sugars, and necessary carbon and nitrogen sources. After about 24 to 28 hours, the culture material in the seed tanks is transferred to the fermentation tank. The fermentation tank is a larger version of the seed. It is filled with the same material as the seed tank and also provides an environment that stimulates growth. The microorganisms continue to grow and multiply. Here, they also excrete the larger quantities of the antibiotic. A continuous stream of sterilized air is pumped into the fermentation tank. Anti-foaming agents are periodically added and, since pH control is vital for optimal growth, acids or bases are added to the tank as necessary. The next step is isolation and purification. After 3 to 5 days, the maximum amount of antibiotic will have been produced, and the isolation process can begin. Depending on the specific antibiotic produced, the fermentation broth is processed by a number of purification methods. The antibiotic is separated and dissolved from the culture using various organic chemical means. At the end of this step, a purified powdered form of the antibiotic is the result. The last step is refinement. This is the step in which the antibiotic can be produced into different applications. Depending on the final form of the antibiotic, various refining steps may be taken to create the desired antibiotic form. We have reached the end of our lesson on bacteria used in the production of antibiotics. But before we finish, let us do an activity. Students, fill in the blanks with the appropriate term to complete the sentences.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Hello again, everyone. How did you do? Let us review the activity together. The sentences should read as follows. The process of fermentation is required to produce antibiotics. This process requires 4 steps. The first step is to start the culture. Before fermentation can begin, the microorganism must be isolated and grown. Once the culture is large enough, it is put into a fermentation tank. This is where the microorganisms excrete antibiotics. Once the maximum amount of antibiotic is produced, it is isolated and purified. The last step is refinement. This is the step that turns antibiotics into different product forms. Did you fill in all the blanks correctly? Great work today, students. This brings us to the end of our lesson. Today, we learned how bacteria are involved in the production of antibiotics. We studied a brief history on the discovery of antibiotics, and we discussed the manufacturing process. Until next time, thank you, teacher. Thank you, students.